Hey you all, I think I will go home early today.
Oh, why you go home early?
I have a learned message.
Okay, see you soon.
Bye.
What happened? Oh, I'm so scared.
Isn't this Nampon's project? Where is she?
It's so hard.
You! All four of you! You were here yesterday with her.
No, it's not me.
You are suspect. Since you don't talk what happened yesterday, I will have to find out the hard way. Let's see which microorganism present here on Nampon station is the same as you all.
What method we do?
Illumina sequencing technology leverages clonal array formation and proprietary reversible terminator technology for rapid and accurate large-scale sequencing. The innovative and flexible sequencing system enables a broad array of applications in genomic, transcriptomic, and epigenomic. The Illumina sequencing workflow is composed of four basic steps. Step 1. Sample preparation. Step 2. Cluster generation by bridge amplification. Step 3. Sequencing by synthesis. Step 4. Data analysis.
Step 1. Sample preparation.
There are numbers of different ways to prepare samples. All preparation methods add adapters to the ends of the DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as sequencing binding sites, indices, and regions complementary to the full cell oligos.
Step 2. Cluster generation by bridge amplification. Clustering is a process where each fragment molecule is isothermally amplification. The flow cell is a glass side with length. Each lane is a channel cut with a round, composed of two types of oligos. Hybridization is enabled by the first of two types of oligo on the surface. The oligo is complementary to the adapter region on one of the fragment stages. A polymerase creates a complement of the hybridization fragment. The double strand of molecule is denatured and the oligo template is washed away. The strands are coronally amplified through bridge amplification. In this process, the strand flows over and the adapter region hybridization to the second type of oligo. The first cell polymerase generates the complementary strand, forming a double-strand bridge. The bridge is denatured region into side strands copy of the molecule that are generated to the flow. The process is then repeated over and over and occurs simultaneously of millions of cluster regions in clonal amplifications of all dense fragments. After bridge amplification, the reverse strand and create and washes off, leaving only flower stand. The three palm ends are blocked to prevent unwanted priming.
Step 3. Sequencing by synthesis. Sequencing begins with the attention of the first sequencing primer to produce the first read with a cycle fluorescent tag nucleotide complete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the attention of each nucleotide, the clusters are excited by a light source and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the lead. The emission width length along with the signal intensity determine the base call for a given cluster. All identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 lead primer is introduced and hybridized to the template. The lead is generated similar to the first read. After completion of the index read, the read product is washed off and the three palm ends of the template are deprotected. The template now flow over and build the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerase exchange the second flow cell oligo forming a double strand bridge. This double strand DNA is then initiated and the three palm ends are blocked. The original forward strand is kept off and washed away, leaving only the research strand. Read 2 begin with the introduction of the read 2 sequencing palm. As with read 1, the sequencing steps are repeated until the deserved read lasts in achieve the read 2 product is then washed away.
Step 4. Data analysis. This entire process generates millions of reads representing all the fragment sequence from pooled sample library are separated based on the unique index introduced during the sample preparation for each sample read with similar stage of base calls. Are locally clustered forward and reverse read are pair creating contiguous sequence. These contiguous sequences are aligned back to the reference genome for variant identification. The pair and information is used to resolve ambiguous alignment.
This gene is not the same my virus.
Not it too. And we too. Not my it too. And we too.